scale. Jahanavi, Harshita, Aditi has to join. Nobody is replying.
Yes, sir.
All
Aditi
of
need to join.
She has joined. All the four have joined. Shall I open like this only? Or shall I open it to the full scale and talk like that yesterday's? You can stop me whenever actually you feel bored or getting actually confused. Okay? You can put questions whenever you want.
Okay, sir.
Shall I stop?
Yes, sir.
Okay. Sorry. Again, it has gone. Hello?
Yes, sir.
You are able to see? Shall I continue to talk? Yeah. I just summarize in few minutes what we were doing yesterday. Simply, it is simple thing is for nearly a century, we are living with what is called ATP as the simple molecule, which is actually promoting the cell's function. Right? Now, the story of actually lactic acid and pyruvic acid started in 18th century when I've given you this thing. What happened to this? Okay. I'm going to the fifth slide. I am going to the fifth slide.
Okay, sir.
Earlier we have discussed, because this is important, the present models are based on Pasteur's experiments on alcohol and subsequently the discovery of thermostable cofactors by Harden and Young. Am I right? And NADH. The model says, present model of intermediate metabolism says that the main function of respiration is catabolism. This is contrary to what actually Lavoisier has defined. He has defined that besides burning the fuels, oxygen plays a role in biosynthetic activity. A cancer is excessive production of cells that involves actually biosynthetic activity. That fundamental rule has been ignored in building the present models. Now, the second important thing is lactic acid was discovered by Scheele in 18th century. Am I right? Similarly, pyruvic acid was also discovered in 18th century, but in vegetables as an unknown acid. And again, that was identified by Berzelius in 19th century, in the first quarter of the 19th century, as pyruvic acid. But throughout 19th century, we never discussed, there was no discussion on pyruvic acid. It was only lactic acid that was actually discovered. And lactic acid was examined from 2 points. Number one, from three research areas. Number one is muscle contraction. Sorry, before that, actually, it is the pathology. People who were suffering from leukemia were shown to have lactic acid circulating in the blood, but lactic acid accumulated when death occurred in their bodies. Okay? Similarly, somebody in the middle of 20th century, 19th century stated that, if you deprive oxygen, lactic acid accumulates. Fletcher, at the end of the 19th century, demonstrated, optimized actually the lactic acid estimations and also optimized the chemographic experiments, and then there they have showed that oxygen supply promotes carbon dioxide. Oxygen, once it is withdrawn or rigor mortis develops, lactic acid accumulates. So accumulation of a lactic acid is considered to be a symptom of death or rigor mortis or fatigue, whatever you call it. Okay? Now, this particular step also requires whether lactic acid produced in the living organisms when they were circulating, is it oxidized? This question came, and the answer came only after 1960s when, who is that man? Okay, I'll come to that thing. Shall I repeat this?
No, sir, we understood.
You understand this thing, okay? You understand that these are the people who started this thing. I have also told about thermostable cofactors. Am I right?
Yes, sir.
Okay, I move on to the next slide. I don't know, this styling is not working actually. Well, this is an important area. This is the important slide where actually the pyruvic acid came into the picture, right? Where here there are two people who won the Nobel Prize in 1929, and these people, Harden and his student Young, have discovered actually thermostable cofactors, and Euler also worked on these areas. Most of his work is in Germany. He was from Sweden. Warburg also worked with Euler. Initially, the discovery that the thermostable cofactor contains phosphate reducing sugar and a nucleotide, that is nicotinamide, is demonstrated by these people, Euler's people. Warburg was part of this thing. There was just a paper which I came across. I am not getting that paper again. There is a difference between actually this thing and this. They are, the Harden and Young equation is different, and then Euler's equation is different. That is important in our modeling. Okay? And what Harden and Young said is two molecules take two phosphate molecules, produce two alcohol and two water molecules as well as two carbon dioxide molecules. Contrary to that, Euler stated that, there's monophosphate that is formed and that monophosphate splits, and that split monophosphate. Now what happens is the energized triose phosphate, which is split from that phosphorylated hexose phosphate, condenses to form what is called Harden ester or hexose diphosphate, was his theory. They are important to remember. Okay. Then we go for, these are the present models of, you are able to see this? These are the, you have seen the PPT, am I right?
Yes, sir.
In that case, you have actually, I have earlier also shown this thing. 90% of the present models depend upon this particular enzyme aldolase, right? It says that the entire glucose or glycogen molecule is reduced to what is oxidized or phosphorylated to hexose diphosphate, which splits into triose phosphate, which comes as a glyceraldehyde phosphate, and oxidation of glyceraldehyde by NAD dependent phosphorylation produces a molecule which is known as 1,3-bisphosphoglyceric acid, and to regenerate the NAD, to increase the flux of glucose is what is actually thought. That is Warburg effect, where he has stated that oxygen inhibits this step, and then oxygen inhibits pyruvate entry into the mitochondria, and then he introduced the aerobic glycolysis on that. But they ignored Havelock Charles' demonstration that crystalline sugar, fructose, is the principal molecule which produces the alcohol. Other sugar molecules like mannose and glucose need to be transformed into that. Today we have glucose fructose. Whenever you take the sugar, invertase in the gut automatically splits it into a glucose and fructose. And this glucose fructose can be exchanged internally also by glucose phosphate isomerase, which is a reversible reaction. In addition to that thing, you also have, this is your assignment, you also have what is called sorbitol pathway, right? Whenever you have a leisure time, spend 2 or 3 days reading that thing's orbital pathway, prepare a PPT, and then present it to me in the next time. Okay? Orbital pathway also gets activated in the gut and also in the internal environment. Okay? That requires actually NADP shuttling and other things. Where exactly it functions, I think Pankaj from Hyderabad University is examining that particular portion, right? Next, next we move to, well, in this slide only, I forgot to tell you one thing. In this slide only, you have, this is the D portion. This is the central step where fructose, phosphorylation to fructose 1,6-bisphosphate or its reversal to fructose 6-phosphate. What Harden and Young and Euler stated is stability of fructose 1,6-bisphosphate produces alcohol. If this stability is destabilized, then what happens? They have not defined it, but from Pasteur's experiment, we know that once alcohol fermentation is suppressed, that is suppressed by lactic acid production, and that lactic acid production converts the globular yeast cell to filamentous yeast cell. Remember? Understood?
Yes, sir.
Then we go to the next step, which I am asking, which I am actually showing here. In this case, Meyerhoff, I told you, in 1934, along with this thing, discovered phosphoenol pyruvate. In 1938, he has stated that pyruvate kinase is a irreversible reaction. It produces ATP on one hand and pyruvate on the other hand. But in 1945, there was an article by Ziegler where they have stated that pyruvate kinase does not produce actually pyruvate, it actually phosphorylates pyruvate to phosphoenol pyruvate. Second important thing is 1945. During that period, during Hitler's period, Meyerhoff was moving between France and USA, and afterwards only he has settled in USA. It took some time for him. But in 1945, he visited this particular Harden and Young equation. Earlier periods, Warburg group and also Meyerhoff group have supported actually Harden and Young equation, the two equation theories. But in this particular case, Meyerhoff came out with one important observation. The observation was there is an enzyme called apyrase. This apyrase hydrolyzes ATP, and that is an important one. In 1941, Lippmann has stated that phosphate exchanges take place when phosphate exchanges between the organic molecules are catalyzed by ATP hydrolysis. That is exchange of phosphate to ATP, the terminal phosphate to organic molecule. It could be a glucose, it could be a glyceraldehyde, it could be any other organic molecule where actually phosphate, terminal phosphate transfer takes place between the organic molecules only when ATP is utilized there, right? And for this, he agreeing with these things, he has stated that any residual ATP after alcohol production by yeast will be incorporated back into the glucose to phosphorylate it to increase the hexadiphosphate levels. But when ATP accumulates inside the cell, ATP is exported outside. So, in other words, ATP accumulation in the cells is toxic phenomenon, right? So, that is exported, and we have what is called ENTPD1, which is actually an extracellular membrane binding, ATP binding protein, which is tethered on the endoplasmic reticulum. The endocytes, that is, sorry, no, I'm sorry, that is red blood cells, right? That is endothelial cells, as well as in case of what is called the lymphocytes, macrocytes, etc., where ATP is hydrolyzed, adenosine is produced, both, all the three phosphates are hydrolyzed, and they will be increasing the phosphate levels in the microenvironment. I have already told you about the EMT. All the cells are migrating into the microenvironment. That includes macrophages, lymphocytes, right? And also epithelial and endothelial cells. All of them will be doing it. And depending upon the nature of the tissue, whether it is a carcinogenic tissue or diabetic tissue, the angiogenesis will be increasing or decreasing. Similarly, lymphoblasts, myelocytes, etc., they will be differentiating into different cells, producing hemoglobin, myoglobin, depending upon the type of tissue, etc. So microenvironment plays an important role. Although Pasteur was not explicit, he realized that biosynthetic cells transform morphologically into a globular structure, that is, they lose their filamentous structure, adherent structure was clearly demonstrated by that man. Then comes, we go to the next slide. Are you able to see that? Are you able to see
Sir,
what happened? Are you able to see? See the
no, sir.
PPT? No?
No, sir. Now we can't see.
Able to see now?
Yes, sir. Now we can see.
Yeah, you have some Harden and Young's experiments, and also from this thing, alcohol production is the primary this thing in the fermentation process. There was also one question when actually Harden and also Euler were discussing about this. The question was how is actually inside the yeast the alcohol is produced. Then Euler demonstrated that there is an autolysis. Autolysis is autophagy type of mechanism. Okay, that actually dissolves the cytoplasmic membranes, and thereby there is a plasticity. The anchorage or cell surface proteins which normally take nutrients from outside are totally blocked, and thereby what happens is internally the membrane system undergoes what is called hydrolysis. This hydrolysis, particularly the membrane hydrolysis, produces the glycerol molecules on one hand, and also on the fatty acid molecules on the other hand. But before that, it is the fructose molecule that is actually stimulating the fermentation process. It is associated with sodium uptake also. Sodium and phosphate are taken against the glucose molecule. So in this particular case, what happens is fructose is actually hydrolyzed, and you have actually this particular pathway that is this thing. According to the present models, it is thought that they go up to the pyruvate. Pyruvate is reduced to lactic acid. Lactic acid is decarboxylated to acetaldehyde, and that produces actually the alcohol is the theory. But it was very clearly demonstrated it is only dihydroxyacetone phosphate dependent decarboxylation that introduces both ethanol and lactic acid, which was shown by Euler and others also, and also particularly Fletcher in 1907 review, he has clear categorically stated that it is down ethanol and lactic acid are produced parallel to one another. It is not one after the other. Okay, they are produced parallel to one another. So this particular step does not occur. I believe you studied the article on moonlighting enzymes. Did you?
Yes, sir.
In that moonlighting enzyme of Celesta Simon, 3-phosphoglycerate, once it is accumulating, it's, it will not convert 3-phosphoglycerate into 2-phosphoglycerate as it is taught in the conventional glycolysis model. What does it do? It goes and inhibits the second enzyme of the pentose phosphate pathway. What is the second enzyme? 6-phosphogluconate dehydrogenase. What does it do? It converts 6-phosphogluconate, which is a 6-carbon molecule, to 5-carbon molecule ribulose 5-phosphate by liberating carbon dioxide. Okay, initially they thought that that is a second mechanism of actually fermentation process where carbon dioxide is evolved. but carbon dioxide produced during the alcoholic production comes only from that particular point, needs to be understood now. Okay. Second important thing is this particular pyruvate kinase has a reversible effects. There are also pyruvate metabolism is a highly confused metabolism on the present models, but it has multiple roles to play. We will come to that step by step. So, have you understood this slide? Yes or no? At least one reply.
Yes, sir.
You understood. Shall I move to the next?
Yes, sir.
Well, this is the partitioning of actually Higgs shuttles and the pentose shuttles and the triose shuttles. What is actually happening is fructose 6-phosphate shuttles play a critical role in regulating actually the other metabolism also. And this metabolism, the fructose 6-phosphate in the presence of glutamine is shunted to hexosamine biosynthetic pathway. And you see that fatty acid oxidation to acetyl CoA, glutamine, which is actually a protein metabolite, are required for this particular pathway activation. It culminates in what is called O-glycosylated and N-glycosylated molecules. These O-glycosylated molecule, one of the important molecule is 4F2HC. This 4F2HC is a co-transporter both for cysteine uptake and also for essential amino acid uptake. But in metastatic cells, this particular molecule is inhibited by the TGF beta, which actually activates actually reprogramming in the stem cells by depositing the collagen molecule on the surface of these molecules. I have already told you that it is the fumarate that is acting as a electron acceptor. We will discuss that further. But for the present, you will see that the confusion on the lactate, why oxygen containing lactic acid is also produced. And that is produced in cytoplasm even according to the Warburg. If Warburg is right, Warburg is right only to that extent. Okay. In this case, what happens is lactic acid enters into the cells through a basigen molecule which is attached to the monocarboxylate transporters, MCT1. There are several monocarboxylate transporters, but two of them are important here, MCT1 and MCT4, I believe. And that actually basigen molecule which binds to actually MCT1 promotes lactate uptake by the cells. This lactate by LDHB is converted to pyruvate. Pyruvate carboxylation in the cytoplasm leads to the production of oxaloacetate. Oxaloacetate phosphorylation gives rise to what is called phosphoenol pyruvate, right? And that actually activates. In addition, what happens is this is actually producing fumarate also. This fumarate oxidation to succinate is important step. And this succinate oxidation has two roles. Succinate can bind to the nucleotides, to the as an epigenetic molecule. It can bind to the chromatin material and activate transcription of specific genes also, or suppression of the specific genes also. Similarly, fumarate also is an epigenetic molecule. Fumarate acts as an oncometabolite also. Fumarate also acts as an oncometabolite, and this oncometabolite plays a critical role in actually in maintaining actually mesenchymal cells. Have you understood?
Yes, sir.
Now, succinate epigenetic modification alters actually the gene expression data, and then succinate oxidation actually activates respiration. Succinate oxidation is SDH. Okay. Epigenetic modifications, you need to understand that, there are several epigenetic modifications are acetylations, methylations, demethylations, and then succinylations, acylations. These are some of the modifications which alter actually the gene expression systems. They bind to a specific genes and then alter, either suppress their expression or activate their expression. That is important mechanism. Now, fumarate is electron acceptor under the anaerobic condition. It is a byproduct of actually arginine and aspartate metabolism. It is also actually activating the succinate production by its reduction, and that reduction is important in activating this thing. 1925, Kaline identified cytochrome C. You know electron transport chain, I believe. Do you know?
Yes, sir.
That is complex 1. Complex 1 transfers its electrons through coenzyme Q to complex 3, and complex 3 will transfer through cytochrome C to complex 4. That cytochrome C oxidase is complex 4, right? Remember that? In between, you have what is called coenzyme Q. According to the present TCA cycle model, citrate is produced in the TCA cycle. It is exported outside, and outside ATP citrate lyase will be breaking down actually citrate into acetyl CoA and coenzyme A. But bulk of the acetyl CoA, acyl CoA, fatty acid oxidations, cholesterol dependent phytonic acid, several of the fatty molecules, including the branched chain amino acids like leucine, valine, isoleucine, they are oxidized only in peroxisomes and they produce what is called bulk of acetyl CoA and also succinyl CoA. In addition to that thing, you need what is called dihydrolipamide for actually activating these CoA transfers, acetyl CoA or acyl CoA transfers from one molecule to the other molecule. That is an important step taking place in cytoplasm, which are associated with mitochondrial systems. Okay, mitochondria associated membranes and vacuoles. Now I move on to the next step. I have already told you the fluxes between actually the fructose 6 phosphate and this thing, they are actually activating, they have two functions. Stability of the hexose diphosphate produces, keeps the dihydroxyacetone phosphate stable, and this in turn will be producing alcohol. The second important thing is if fructose 6 phosphate is shuttled, if because of actually destability of this particular thing and dephosphorylation of fructose 1,6 bisphosphate to fructose 6 phosphate, accumulation of fructose 6 phosphate picks up what is called the amino acids, and there the glutamine plays an important role, and then it activates actually a series of steps which culminate in N glycosylation and O glycosylation, which I have already discussed. Whereas the plasticity or membrane hydrolysis produces what is called the fatty acids, the free fatty acids binding to the GPCR receptors. There are 800 GPCR receptors, out of which hardly 20% of them alone are characterized. If you characterize new GPCR, you will be a Nobel laureate. Okay. Now, in this particular case, the free fatty acids, short chain fatty acids bind to different GPCRs, long chain fatty acids bind to different GPCRs, hydroxycarboxylated molecules. There are three of them. One is lactic acid, right? It is a 3-carbon carboxylic acid. Am I right? Am I right? CH3COCOOH. What is it? It is a carboxylic acid having hydroxy group on alpha carbon atom. Am I right? Yes or
Yes, sir.
no?
Yes, sir.
Now, in a similar manner, beta hydroxy butyrate, octanoic acid, which is a 8-carbon hydroxy carboxylic acid, this beta hydroxy butyrate can be produced from branched chain amino acids, but octanoic acid is actually partially hydrolyzed fatty acid. That too, arch chain fatty acid that produces hydroxy. These three receptors bind to what is called a specific type of GPCRs, and these GPCRs are what are called as HCARs. I'll come to that little later, but for the present, you understand that binding of that particular molecules have a differential effect, and free fatty acids bindings can also activate what is called the G alpha or G beta or G zero type of actually internal molecule, and that molecule will be actually activating cyclic AMP dependent protein kinase A, and that way it will be activating hydrolysis of actually these things, and that leads to production of second messengers in the form of inositol phosphate, in the form of calcium, calmodulins, protein kinase C, diacylglycerols, etc. All of them act as a secondary messengers in activating further metabolic pathways in different ways. But the glycerol, which is byproduct of this membrane lipid, is first oxidized by what the FAD dependent, cofactor dependent, FAD cofactor dependent this thing which has association both with the membrane and also cytoplasm, and that produces dihydroxyacetone phosphate. The enzyme triose phosphate isomerase produces glyceraldehyde 3-phosphate. One thing which we need to know here is the first part of the G1 phase of the cell cycle. I think yesterday I explained that contains what is called cyclin D1 and also P16, P14, P15 proteins only. And that particular portion is active during the mRNA synthesis. Then P53 also enters into the picture. This P53 activates P21. Hitherto it was called as inhibitor, but it has additional function also. P21 activates actually the P21 activated kinase back. P21 activated kinase, and that activates actually 3-phosphoglycerate dehydrogenase, that is PGK1, sorry. Phosphoglycerate kinase will be activated allosterically by that thing to increase the ATP production in this thing. I have already told you that this particular pathway is non-existent in proliferative cells. It is activated only when lactate enters into this thing. That is why in COVID, in diabetes, where lactate uptake is minimized, this particular pathway is not active. Metformin, which inhibits actually the lactate uptake and also lactate exit, will not allow actually this particular pathway to be activated. That is the reason metformin is also considered as anti-cancerous drug apart from diabetic drug, but it also has a side effects because it inhibits actually the cholesterol biosynthesis and other biosynthetic pathways. Totally MPC1 and MPC2 are also inhibited by this particular system. Okay, then I move on to this thing. Well, I told you already the central pathway There's one pathway which I come to that thing, that is cysteine biosynthetic pathway, and that cysteine biosynthetic pathway. Even for the activation of this PSAC in serine biosynthetic pathway, you require pyridoxal phosphate. The aminotransferase requires pyridoxal phosphate. Pyridoxal phosphate is a component of vitamin B, isn't it? It is vitamin B6. So unless vitamin B6 or carboxylation comes into this picture, you will not have this particular pathway activated in this. For that, what happened is metastatic cells, they require purine also. That is, mRNA also contains what is called thymine pyrophosphate, and also the adenine and guanine, these things. For that, you require methylation of uracil, and that particular step requires activation of this thing. So, it has adopted a second mechanism in which actually the serine biosynthetic pathway is activated, and that serine biosynthetic pathway is this thing. I stop at this level and then go to parallel pathways which are activated by insulin. Okay, insulin in 19th century only, Minkowski stated that pancreatic islets play a critical role in actually producing insulin. Right? If you ligate pancreatic duct along with the nerve, it will actually hydrolyze the exocrine glands first. Endocrine glands will be activated. That was stumbled upon by what is called this man, where I am forgetting MacLeod. Along with MacLeod, two people got the Nobel Prize, isn't it? Banting. Banting actually was working in the army. During that period, he came across this paper, but he was not having laboratory facilities. Therefore, he went to MacLeod, and MacLeod provided him, although he was skeptical about the mechanism, he provided him the facility, and this facility actually he utilized. He gave him another assistant, Best, and he was following exactly what is happening. So he was providing funds, he was he has given the placement, but also guiding them as to how. Banting had no experience in experimental laboratory, and at the final stages, when insulin purification came into the picture, he has to recruit another person, that is, I think, Colin. In that man actually got it. When the Nobel Prize was awarded, Banting misbehaved with MacLeod, and MacLeod should not get, Best should get, Best and I should get, because they did the experiments. Well, that is a social problem, but basically, what happened? MacLeod Nobel lecture highlights this particular portion. So Banting's lecture will not highlight that. Now, this particular portion here stated that insulin injection promotes PI uptake by the from the blood by the tissues. PI disappears from the blood when insulin is given inside it to you. Where is that PI going? It is going into the tissues. What is it doing? It is also activating hexose phosphate shuttles. That is what MacLeod reported. In addition to that, he has also reported two other things. Hyperglycemia is a protective agent, but it is a pathogenic agent. It is inflammatory causing, but it will not kill the organism. But if the patient enters into hypoglycemia is lack of proper sugar circulation in the blood. Then the patient enters into the coma and ultimately dies also. During the discovery and purification of insulin, Allen another medico, he was working on actually the starvation diets. He has stated that, starvation diets cured. Jocelyn was one of the senior physician during that time. Alan and Jocelyn put together. Jocelyn gave credit to Alan only because it was Alan's hypothesis. And Alan and Jocelyn, they have demonstrated that, starvation diets can improve several diabetic patients. But some of the patients were dying very early, earlier than what naturally they were dying. They were confused, but because of the advantage of the starvation diets, some people started taking the risk. Anyhow, we are dying. Let us take the risk. That was the principle in that. But in 1936, Hemsworth came up with the hypothesis that diabetes patients can be distinguished into two categories. Number one is insulin sensitive, where insulin is absolutely required for their for reducing the sugar molecules in the blood. But and insulin resistant, that means even if insulin is given, some people will not respond. There you need to maintain actually the dietary maintenance, exercise, lifestyle changes which play important role in this, in maintaining the health of the individual. Have you understood this thing? Have you understood? Have you understood this thing, Janavi?
Yes, sir. Yes.
And during this period, Hemsworth's work was taken up later on after nearly 50 years by Revan. Revan, here there are lectures in honor of this particular Nobel laureates arranged by several institutes. He has introduced the term syndrome X. Syndrome X is a conglomeration of several diseases. Yes, yes. Okay, shall we stop at this level? Yeah, I will. I will stop at the second this thing. One more slide I'll explain, and then leave it there. Okay. Revan introduced actually syndrome X, which is a conglomeration of several diseases, which include the heart attacks, vascular disorders, cerebral disorders. This he called it as a metabolic syndrome. If you hit Google metabolic syndrome or syndrome X, you will get actually the cluster of diseases which are assembled under this syndrome X. He has stated that insulin resistance is the cause of this thing, which results in the glucose intolerance, hypertension, dyslipidemia, etc. Heart attack also is part of this particular segment. Well, why we are not able to understand the diabetic theories and then this thing. I told you Minkowski discovered that ligation of this thing and then removal of the pancreas will control actually blood sugar level. But to maintain the blood sugar level, you actually the what you call place the pieces of actually the pancreas isolated, grab them into abdominal regions of that particular dogs. Then the dogs will survive, and then hyperglycemia will be reduced. So something that is secreted in the pancreas islets, not the pancreas proper. That is, exocrine glands are not having any role, but endocrine glands have a role in secreting a hormone which maintains blood sugar was his principle. That is on which actually Banting and MacLeod also purified the insulin proteins. Fluger, who was by that time a Nobel laureate, he was running an article on a journal which is known as Fluger Archives. Even now, you have Fluger Archives. In that Fluger Archives, he published Minkowski's articles as they have recommended honestly, but he questioned actually the results. What he has stated is the carbohydrate metabolism is maintained by liver glycogen synthesis and glycogen breakdown. Glycogen breakdown in the liver circulates in the blood, increases the glucose level. So hyperglycemia is because of that. Fluger's theory was the duodenal portion. Are you able to see this? The duodenal portion, right? This is the stomach, and then this particular portion is the jejunum portion. Duodenum enters into this jejunum, and the jejunum continues with the ileum, and ileum you have a rectum where actually the fecal matter accumulates and then eliminated. Now, bariatric surgery, this jejunum portion is removed. Nothing will happen to the patients. What happens is actually the fat that accumulates in the abdominal region. Women after the delivering the children or people who are having actually the diabetic symptoms, they have abdomen, the fat, visceral fat growing enormously. The reason for that is the mesentery region in this particular intestine contains several of the adipocytes. Adipocytes can store enormous amount of fat. There is no limitation. In all other cells, you have the limitation of this thing. But hydrolysis of the lipids leads to the inflammatory processes again. So if you limit your nutrients, you are limiting actually this particular visceral fat. The reason here is here once the food enters into the duodenum, liver secretes bile which converts acidic food into the alkaline food. Second important thing is it converts actually pancreas secretes pancreatic amylases, pancreatic lipases, pancreatic proteases, trypsin, chymotrypsin, which break down actually peptones and shorter peptides into to the two carbon and one carbon amino acids and thereby liberate amino acids into this thing. The present model only says that carbohydrates are oxidized and carbohydrates are source of the lipid. That is another fundamental error in the present model. And because these amino acids contribute to this thing, to the carboxylation by MPC1 and synthesis of citrate and lipogenesis, etc. We will discuss that too when we discuss actually the Krebs cycle associated with lactic acid entry and also pyruvate metabolism. Tomorrow, will you be in a position to meet?
I'll let you know, sir. I'll let you know by tomorrow afternoon.
Yeah, yes or no, you tell me. If you are in a position to actually come out tomorrow, we can continue it at a faster rate. Afterwards, I'll give you some experimental tools on which you need to actually have a thorough knowledge of this thing. Hyderabad
Okay, sir.
University and other places, I was asking actually whether practical training can be given to you people, but there are charges. I don't know whether you will be in a position to pay that or not, but each candidate for a shorter this thing like what is called NMR analysis or gene analysis or Western blotting, etc., they require money, and that is slightly heavy and you require that is what is called around 35,000 rupees or so for 3 months summer holidays. Whether your college gives summer holidays or not, that you think about it. My role is only in actually giving you the theoretical input, training you on the theoretical side. Are you in a position to distinguish actually the present model of metabolism and the model which I am trying? to explain?
Yes, sir.
Okay, thank you very much.
Thank you,
Okay.
sir.
Are you meeting tomorrow? You mail me in the morning hours so that I'll prepare some of the slides, organize them because there are several things, several questions are there in my mind. I'll be examining several simultaneously, several questions coming from my friends and other scholars also. And therefore, I require time for this particular classroom because you are at the fundamental level, or I am trying to go to the fundamental level and trying to explain you. I don't know whether you are following it or not. That is your problem. I have given you today one assignment. Try to complete it at the earliest and then come back. Okay?
Okay,
Yeah, Jahnavi, Shriya, Harshita are there. Aditi is not there today.
yes,
She
sir.
is tired. She is
Yes, sir.
tired.
She said
Tired of.
she'll be attending the other class.
That's okay, but discontinuity gives you. Already we are discussing actually a new theories, and these new theories, if discontinuity is there, you will not be in a position to follow up. One unless you exchange and then discuss among yourself and share it.
Okay,
Normally,
sir.
the tendency of the student is I learned something, I keep it myself, as if it is a secret purse that is doing it. That is what research scholars also do, but all of them do the same stupid thing. Okay, same Warburg effect. They run around actually TCS cycle, glycolysis, etc. Jealousy is not important in this thing. Sharing the knowledge improves the knowledge. There is what is called dialectic exchange of ideas that opens up in this thing. In my first webinar lecture, I was telling the organizers, you liberate the literature, the cost of research will go down because some thinkers will help in updating and modifying the present erronic, error prone pathways which are remaining as a dogmas. They agreed with that thing, and then slowly they are liberalizing it also. Thank you very much. Okay, I I am closing.